In the process of transcription, the main goal is synthesizing messenger RNA from a DNA strand. In the most basic terms, an enzyme called RNA polymerase prices two strands of DNA apart and joins together RNA nucleotides complementary to the DNA template strand, thus elongating the RNA polynucleotide. During the first step of transcription initiation, an RNA polymerase binds to the promoter of the DNA strand. In a prokaryote, the RNA polymerase is able to bind to the DNA by itself, but in eukaryotes, a collection of proteins called transcription factors mediate the binding. An example of this is a TATA box, a block of DNA filled with T and A nucleotides that signifies to the RNA polymerase where the starting point is. Once connected, the polymerase moves downstream, unwinding the DNA and elongating the RNA transcript from the 5-strand to the 3-strand. After this, the DNA strands reform in a double helix. During this elongation behind the polymerase, the RNA peels away from the template strand. Eventually, the RNA transcript is released and the polymerase detaches from the DNA. In eukaryotes, RNA polymerase II transcribes a sequence, which codes for a polyadenylation signal sequence, which codes for termination.
Once the mRNA strand has been made, enzymes in the eukaryotic nucleus modify it in specific ways before the genetic messages are dispatched to the cytoplasm. Before it leaves the nucleus, the 5' strand received a modified form of a guanine nucleotide and the poly A tail at the 3' end. Also, in the nucleus, the mRNA undergoes a process called splicing, where a structure called a spliceosome performs a cut and paste job on the strand. Although the mRNA strand contains roughly 27,000 nucleotide pairs, it only takes 1,200 nucleotides and RNA to code for a protein. Hence, most of the nucleotides, introns, remain inside the nucleus, while the ones that actually code for the protein are called exons, as they exit the nucleus. The catalytic component of the spliceosome are ribozymes, RNA that acts as enzymes. Once the splicing is complete, the strand of mRNA is transported out of the nucleus and the process of transcription is completed.
During translation, the goal is to read the genetic information of a cell and build a polypeptide accordingly. Before considering the process, it's important to understand the function of transfer RNA, or tRNA, an RNA molecule strand that is only about 80 nucleotides long. On one end of the tRNA is a sequence of 3 nucleotides that match up with this sequence of nucleotides of mRNA. This is called the anticodon side, and depending on the matching codon on the mRNA strand, it matches up for a specific amino acid. Because all 20 amino acids are stocked throughout the cytoplasm of the cell, the tRNA is able to grab the corresponding amino acid on its other side and bring it to the mRNA strand. The correct matching of codons and anticodon is carried out by aminoacyl tRNA synthesis, a family of related enzymes. The actual matching of codons and anticodons are more lenient than that of codon positions. For example, the nucleotide base U at the 5' end of a tRNA anticodon can pair with either A or G in the 3rd position for an mRNA codon. This flexible base pairing at this codon position is called wobble. During protein synthesis, the coupling of codons and anticodons occur at the ribosome, which consists of a large subunit and a small subunit, each made of proteins and other ribosomal RNAs. The actual structure of the ribosome molecule very much reflects its function during protein synthesis, as it holds 3 separate sites. The P site holds the tRNA carrying the polypeptide chain, while the A site holds the tRNA carrying the amino acid to be added to the chain next. Discharged tRNA leaves throughout the E site. The tRNA and mRNA are held close in proximity by the ribosome and are positioned for the addition of the carboxyl end of the growing peptide. The formation of the peptide bond is then catalyzed. As the peptide becomes larger, it passes through the exit tunnel of the larger subunit of the ribosome. The actual building of the polypeptide is done in a manner similar to the process of transcription, as there is an initiation, elongation, and termination stage. In the initiation stage, a small ribosome subunit binds to both mRNA and a specific initiator of tRNA, and the start codon signals the start of translation, which establishes the reading frame for the mRNA. A larger subunit of the ribosome is added to this equation, and the translation initiation complex is formed. It's important to note that the cell also expends energy obtained by hydrolysis of a GTP molecule to form the initiation complex. During the elongation stage of translation, amino acids are added one by one to the previous amino acid at the C terminus of the growing chain. The elongation is a three-step process, starting with codon recognition, which requires hydrolysis of one GTP, which increases the accuracy of this step. One more GTP molecule is hydrolyzed to provide energy for the translocation step. The mRNA moves through the ribosome in one direction only, the 5' end. The ribosome and the mRNA move relative to each other, codon by codon. In the translocation step, the tRNA in the P site moves to the E site, while the tRNA in the A site is moved to the P site. This continues until the mRNA is finished and the peptide chain has been formed. Finally, during the termination stage, either a UAG, UAA, or UGA codon appears, which codes for stop. A release factor, a protein shaped like an amino seal at tRNA, binds directly to the stop codon in the A site, which causes the addition of a water molecule instead of an amino acid on the polypeptide chain. This reaction breaks the bond between the completed polypeptide and the tRNA in the P site, releasing the polypeptide through the exit tunnel of the ribosome's large subunit. The remainder of the translation assembly then comes apart in a multi-story process aided by other protein factors, which require the hydrolysis of two more GTP molecules. The entire process of creating a polypeptide takes roughly a minute, and as a result, multiple ribosomes translate an mRNA at the same time. Once a ribosome is far enough past the start codon, a second ribosome can attach to the mRNA, eventually resulting in a number of ribosomes trailing along the mRNA. Such strings of ribosomes are called polyribosomes. Although most ribosomes are free in the cytoplasm, some can be bound to the rough ER based on the desired function of the peptide. The polypeptides of proteins desired for the ER or for secretion are marked by a signal peptide, which targets the protein to the ER. The signal peptide, a sequence of about 20 amino acids at or near the leading end of the polypeptide, is recognized as it emerges from the ribosome by a protein RNA complex called SRP. Once recognized, the SRP can take the ribosome complex to the desired part of the ER, and the signal peptide is removed by enzymes. This completes the process of translation.